Okay, so I am going to tell you about my thesis work today, which is on Saxicolous lichen biomonitoring of inorganic nitrogen deposition in the southern Sierra Nevada. And to kind of give you an overview of the rundown of what I'll be saying today, kind of introduce you to the issues that we're looking at and the questions we're trying to answer, and the methods we're going to use to answer those questions, and some of the preliminary results that we got from the 2019 data collection, as well as the next steps in data collection that we hope to get to help answer some of these continuing questions, and then what conclusions we've drawn from that. And so this, just to help introduce this a little bit, this is a picture from the Sierra National Forest and the John Muir Wilderness within that, which is right along the transect where I sampled. So the problem or issue that we're looking at using lichens is nitrogen pollution. Probably most of you in botany and plant pathology don't really think of nitrogen as a bad thing. It's an essential plant nutrient, but it can be too much of a good thing for native plant ecosystems, essentially, that are not adapted to high levels of nitrogen inputs from human activity, essentially. So too much nitrogen can be known to cause problems such as changes in sensitive plant community composition. It can alter carbon to nitrogen ratios in trees and contribute to tree death, which is a big issue in the Sierra Nevadas, as well as be implicated in increased wildfire activity, frequency, which is also what California is well known for. And briefly, where does this come from? Well, for California and the Sierra Nevada, the Central Valley is the source. In particular, it's the breadbasket of the country, but definitely for California. And so the two main actual sources for that would be agriculture as well as transportation. There are some large urban centers in the Central Valley. And then in terms of agriculture, the cattle, or not just cattle, livestock waste, as well as fertilizers for crops are big inputs. So kind of introducing you to the Sierras, but in particular high elevation lands in California. There's over 2 million acres of Class 1 wilderness that are above 6,500 feet in California as a whole. And when we're thinking about the southern Sierras, Yosemite and Sequoia Kings Canyon National Parks are also Class 1 areas that fall in that category and are particularly well-known, charismatic places that people like to go recreate. Why do we care about Class 1 areas? What does that mean? Class 1 areas are afforded special protections under the Clean Air Act, and federal land managers are required to monitor those resources, essentially, that fall in those areas. And one of those resources would be air quality. So essentially, we have to monitor air quality in these Class 1 areas. And many of these Class 1 areas, and my study area, are in very close proximity to the Central Valley, to that nitrogen pollution source. So then what kind of monitoring capabilities do we have for these areas, which are often quite remote? The first one are nitrogen deposition mathematical models, essentially, that take data from air pollution monitoring stations and create a model for California, for the entire country, really, depending on which model you look at, and you can get nitrogen deposition estimates that way for whatever area you're interested in. But these models have been shown, one of the problems of these models is they have been shown to be inaccurate in complex topography, which is very much what we're dealing with high elevation areas in the Sierra Nevada. Another method which actually gives you point measurements and actual measurements for an area are these ion exchange resin collectors, which are pictured in the picture on the right there, and they measure bulk deposition of nitrate and ammonium. So those are kind of two of our major nitrogenous compounds that are at play when it comes to nitrogen pollution, but they're not the only ones by any stretch of the imagination. And bulk deposition just means air deposited as well as precipitation deposited. The downside of these types of collectors is that you have to go out, set them up, you know, leave them out for whatever, 6 to 12 months, and then take them back down, and then you have to store that equipment and analyze the resins, and you know, you have to buy all this equipment to create these basically, and there can be problems with them out in the field as well. And so finally, I'm talking about lichens. So there are lichen biomonitoring tools available as well, and typical lichen biomonitoring tools, you know, allow you to go to multiple locations and sample relatively large areas, because most of these types of tools just, you know, require one visit essentially. So yeah, that can be an advantage. Quote unquote typical lichen biomonitoring tools are, you know, we're limited in the Southern Sierras essentially, because these high elevation areas, from what I saw, above about 7,500, 8,000 feet, lack epiphytic lichens in this area, and epiphytic lichens are just lichens that grow on trees. So that finally leads me to define Saxicolous, which are rock-dwelling lichens, and in the Southern Sierras, you can find rock-dwelling lichens or Saxicolous lichens relatively easy and at all elevations. So that's kind of leads us into why we're looking at those. And so most of my research questions, you know, have to do with these Saxicolous lichens because that's what I'm delving into. And in particular, Saxicolous lichen elemental content in relation to nitrogen deposition. So my first question, is there a relationship between nitrogen content of these Saxicolous lichens and then the modeled nitrogen deposition estimates? And then kind of taking that a little bit further, is there a relationship between scores created by principal component analysis for the Saxicolous lichen elemental content and then those nitrogen deposition estimates as well? And finally, does nitrogen content of Saxicolous lichens co-vary with that of epiphytic lichens that are collected at the same sampling sites? So kind of just to give you a, you know, starting into the methods, a little overview of so-called typical lichen biomonitoring tools. Those tools typically use epiphytic lichens, or lichens that grow on trees, as you can see from the photo right there. And so the idea with this tool, lichen elemental analysis, is you collect lichen right off the tree, just like in this photo, stick it in a bag, clean it up later, and then you send it off to a lab that does this elemental analysis, and they send you back a nice spreadsheet with you know, all the concentrations of whatever elements you were looking at, so including nitrogen. And we know from past research that the species of lichen studied, and one of my epiphytic lichens that I will talk about, these lichens are known to accumulate pollutants proportionally to environmental concentration or atmospheric availability. So they're good indicators, essentially, is the message from that. And kind of taking this logic or this research a little bit further, you can also collect lichen and then transplant it in a location that you want to study. So the idea is that you would collect it from a relatively clean site and then transplant it into a site that you want to monitor, likely more polluted or whatever, leave it out for some months, remove, and then do elemental analysis. So a little bit more of a pain, kind of like those ion exchange resin collectors, because you have to make 2 trips. But can be very useful in areas that lichen doesn't exist naturally. Okay, so kind of zooming in on the study area a little bit more, the sampling I did last year in 2019 was at 21 sites along a nitrogen pollution as well as an elevational gradient. You can essentially see Fresno in the lower left-hand corner, and then kind of my sites move to the northeast from there, increasing in elevation. And getting, you know, if you think of Fresno as the pollution source, you're getting farther away from that, you know, the Central Valley, the pollution source essentially. So the elevation of those sites range from 600 to 11,000 feet in elevation, so quite a large range. And I collected Umbilicaria thea, which is this little chocolate chip looking lichen that grows on rock. So this is our Saxicolous lichen. And then Lotharia vulpina, which is our epiphytic lichen, which probably most of you have seen if you've gone to the east side of Cascades or the Sierra Nevadas for that matter, because it's covering the trees basically at middle elevations. So these samples were collected, cleaned up, and then sent to a Forest Service lab for inductively coupled plasma optical emission spectrometry to get concentrations of 27 different elements, including nitrogen. And so we plan on sampling again this summer, pending everything goes normally. And we'll go back to some of these same sites and collect rock samples where the lichens were growing on, as well as lichen community data to kind of supplement and add some more variable data into the mix. And then the other thing we're going to do is set up these ion exchange resin collectors along this, maybe not the same transect, but similar transect, same basic area, with Lotharia transplants to see how those fare, and then kind of intensify that grid of transplants in higher elevation wilderness areas, essentially, that lack these naturally occurring populations, so above about 8,000 feet typically. And so now going into my statistical methods and all that, and also giving you a visual of kind of what these models look like. So just on the right hand side we have a screenshot. Essentially, I should have made a nice map and arc map, but it's a screenshot of the CMAQ model, which is the Community Multi-Scale Air Quality Model, that's essentially showing an overlay of modeled nitrogen deposition in this study area. So we have the black triangles and circles are essentially those same 21 collection sites. I've got Fresno circled in the lower left-hand corner again, so that you can kind of see it's the same basic thing. And high nitrogen deposition is kind of pink purple, and then ranging to this more blue teal that's low deposition. And you can see the gradient somewhat easily that way. It theoretically gets cleaner as you move to the northeast or just east. So then using the CMAQ model, as well as the lichen elemental analysis data, I also used RStudio for regressions and PC-ORD for principal component analysis. Bruce did the principal component analysis because I don't know how to do that yet, so thanks, Bruce. And yeah, so just to give you a little review of our first question, is there a relationship between the nitrogen content of Saxicolous lichens and the modeled nitrogen deposition estimates? So we think about nitrogen content in lichens in percentages. So we've got percent nitrogen of Umbilicaria on the y-axis, and then we have that CMAQ nitrogen deposition rate, which is in kilograms nitrogen per hectare per year. And there is a relationship. It's this very interesting curved relationship, and it's a significant one at that, with a p-value less than 0.05. We don't really have variables to explain why why this is doing what it's doing, basically. And actually, to preface, I should have said what we expected to see was more of a linear relationship, and instead of where, as you can see on the scatter plot, where CMAQ deposition is low, we also expect percent nitrogen of Umbilicaria to be low, but there's quite a few points where it's high. And then middle deposition gets low, and then it gets high again at high deposition, which you would expect. And again, we don't have, our data sets are too small for other variables. We weren't able to explain using this regression. And also, just as a kind of side note, the CMAQ modeled nitrogen deposition is significantly correlated with elevation, as well as essentially distance from the pollution source, which would be, you could think of as Fresno or the Central Valley. So the further you get away from that pollution source. So same curved relationship. So you can kind of think of the CMAQ nitrogen as a proxy for elevation as well as that distance. And so then kind of diving a little bit further into kind of the whole elemental profile for Umbilicaria, Bruce was able to do the PCA, and essentially he created these scores that, and we have two separate ones here in these two separate graphs. The one on the left, more associated with crustal elements, or elements that are present in soil and rock, and then the nutrient scores that are more associated with macronutrients like nitrogen, phosphorus, potassium, etc. So you can see on the left-hand one, the crustal scores, we see another curved relationship when we plot these against the CMAC nitrogen deposition estimates, which is on the x-axis, and that is significant as well. I guess I didn't refresh you to my question. So basically, we're just looking to see if there's a relationship between these two things, CMAC and then the PCA scores. And interestingly enough, in the right-hand side, the nutrient-related scores from PCA, which are on the y-axis, don't appear to have a relationship to the CMAC nitrogen deposition estimates, which is pretty interesting. And so I'm definitely not an expert in this by any stretch of the imagination, but essentially the takeaway from this is it's unlikely that factoring out that crustal elemental contribution to Umbilicaria's profile of elements would clean up the CMAC Umbilicaria nitrogen relationship. So still more questions. And then before I kind of go into question 3, our last question, I did collect Litharia, our epiphytic lichen, when it was present. And then, so I also just compared that to the CMAC nitrogen deposition estimate. So we've got Litharia on the y-axis and CMAC on the x-axis, and this has a nice, clear, linear relationship. It is a small data set, but it is a significant linear relationship with the significant p-value and good r-squared value to 0.79. So in some ways, you could somewhat loosely think of Litharia's percent nitrogen as a proxy for nitrogen deposition as well, given these samples. So then, to remind you of my last question, does the nitrogen content of Umbilicaria co-vary with that of Litharia collected at the same sites? Again, it's pretty small sample size, poor r-squared valued, and this isn't significant in terms of a p-value, but you can see kind of the beginnings of that similar curvature to what we saw with the percent nitrogen Umbilicaria plotted against the CMAC nitrogen deposition. So that's interesting, and there could be it would be interesting to intensify that sampling potentially. And so other factors at play that are potentially causing this strange curved relationship. So one of the things I noticed was that some of the Umbilicaria samples, when I pried them off the rock, they had sometimes would have a lot of pollen accumulated on the lower surface. And so I found out later there can be very nitrogen-rich pollen. So we thought maybe that was potentially causing this. Fortunately, I scored the pollen abundance, I guess, pretty well, and we didn't see a relationship there. I collected at all sorts of different topographic positions. So, there's not enough data there to really show any solid trends with topographic position having an effect on the nitrogen data. That's obviously a complex thing when it comes to airflow and movement and all that. And then canopy cover, may have some kind of thermal sheltering effect, or, maybe concentrate nutrients or help leach nutrients off by, keeping moisture in, who knows. But the data set that I have for that of a canopy cover is pretty subjective and too small, so that needs evaluation. Snow burial effects, similar type of thing, sheltering or maybe even leaching of nutrients. Don't have any data on that, still have to figure out if that would be feasible to collect data on, et cetera, et cetera. And then lastly, nitrogen content of rock. Unlikely that there's, high nitrogen in rock, but I have read a couple papers recently that do show some evidence that there can be, some there. So who knows? That's why we're going to collect rock samples as well. And so in conclusion, there's a strong relationship between those umbilicaria samples and the CMAC nitrogen deposition estimates, but we lack the variables to interpret those results. Small sample sizes is definitely a problem with that interpretation. So hopefully additional data collection of the community data, rock samples, Lotharia transplants, as well as the IER collectors will offer more insight. And I'm running out of time, I'm going quickly. So this project is funded by the U.S. Forest Service, as Bruce mentioned, and big thanks to Bruce and the McCune Lab for taking me on finally after all these years, and also Sarah Jovan with the Forest Service and FIA program, and especially big thank you to Trent Proctor and the rest of the Region 5 Air program. Trent, I think, is listening, and it is his first day of retirement, so I just want to thank him and say that if he hadn't hired me 2 years ago, I might not be here exploring this project. So I'm very, very thankful, and this has been a lot of fun. And I'd now be happy to take questions.
All right, Adrienne, thank you. We're open for questions. If you just type question into the chat box, I can just call on you as they appear in the chat box. I'll start out. How sensitive is biomonitoring to levels of fluctuation in nitrogen? So, I mean, temporally, like, so we've seen recently in this current pandemic where we've had due to a big decrease in nitrogen levels and things like that. Are biomonitors able to reflect that? Is the turnover in situ in organism, is it fast enough to reflect those types of phenomena?
I don't want to say outright no, but these are lichens as biomonitors are kind of more useful in a larger time scale. We typically try and think of them in terms of like the annual nitrogen cycle, but also like precipitation cycle. Because of the way that the lichens work, essentially. So I don't know, they're very slow growing. They, theoretically could accumulate pollutants slowly. So I'm not sure, and sites where they revisit to see changes like this, they're typically revisited on 5 to 10 year intervals. So it's kind of a larger scale, time scale thing. So yeah, I don't know how accurate that would necessarily be.
Okay, thanks. All right, Tom.
Yeah, thanks. Great talk. Could this weird curvilinear relationship with nitrogen have something to do with, say, like growth? Could you do biomass? So, I mean, maybe a little bit, it stimulates it so it doesn't accumulate as much, and then it slows down growth and accumulates. You see what I'm getting at? Anyway, I'm wondering if you ever considered that, and if you have some way to look at biomass and see if it's correlated.
This isn't really answering your question, but one thing that I noticed, because again, this was over a huge elevational range, really. When you're at lower elevations with these Saxicolous lichens, these rock lichens, they're much bigger. It's a much less harsh environment. And then you get up into these higher elevations and they're a lot smaller. I mean, I guess you could maybe argue it's like, oh, there's so much nitrogen, they're growing bigger because of that. But it's also just, the complete differences in environment. I can't say that, I mean, it seems like there would be potentially some effect there. As far as I know, no one has really looked at that, and particularly when it comes to nitrogen. I know I think there's been some experiments with phosphorus and showing, adding phosphorus allows for faster growth in lichens, but as far as I know, not nitrogen.
Thanks, appreciate it.
Kyle?
Hey, Adrienne. I have a question regarding, I guess, the specific genus, the umbilicaria. Is there like a potential that there is that nitrogen uptake is just like sufficiently different from Lotharia, or there's a different mechanism of uptake that is not allowing for this relationship to be shown, like just a purely like physiological phenomenon, or is there a reason to expect just by there being, you know, by it being another lichen genus that you would expect similar uptake of nitrogen?
So aside, and I mean, physiology, sure, because they're, you know, totally different species and all that. But one thing that really kind of divides the two is Lotharia is, you know, growing on trees. It's up above that soil level, you know, so just their, yeah, in terms of height above the ground is very different. And so what they're exposed to, the environments they're exposed to, could potentially be really different. Another thing is Umbilicaria is a foliose lichen, which means it's kind of more leaf-like. It's got like a flat surface essentially. And then the Lotharia is a fruticose lichen, and it has, so it's more shrub-like essentially, and has a lot more surface area. It also has like one tiny little point of attachment to the bark that it's on, whereas you know, if you recall that picture, the Umbilicaria is very oppressed to the rock, and thus the pollen accumulation. I've never seen that with epiphytic lichens before. So just straight up growth forms are very different, and you know, higher surface area often equals higher sensitivity with lichens. So again, that doesn't like really necessarily address your question, but I think that when it comes to lichens, yeah, those growth forms can make a big difference.
Ian?
Yeah, I was wondering, kind of somewhat similar to Joey's question, but how does weather affect the fluctuations that you would see in nitrogen levels in lichen at any point in time? Because like I did some similar stuff with moss as a bioindicator in an urban environment, and you kind of see that like rainfall, you know, it kind of equilibrizes what's on the inside and outside of the tissue. So I was just wondering how, you know, if you go sample directly after a rain or during a dry period, how that might change how you're going to see the nitrogen levels.
Yeah, I think it's exactly like you just said, basically. Yeah, directly after a rain could make a big difference in what you see with nitrogen or other elements, because of that potential leaching effect of rain. So typically we try and sample, you know, during the dry season to avoid that, and you know, keep it consistent in that sense. So yeah, I mean, yeah, precipitation has a big effect there. So, you know, keeping those sampling times consistent and just being aware of that's how these organisms work basically is kind of interesting. This particular system, the Southern Sierras, you know, it's pretty dry. Most pollutants are dry deposited, and so there's not as much, you know, leaching that would occur compared to the Pacific Northwest, for instance.
Gotcha. Cool. Thank you.
All right. Any other questions for Adrienne? All right. Well, let's unmute and give her a round of applause, please.